Hello and welcome to Chapter 4. We are starting our next unit, Unit 2, and we are going to take a look inside the cell. Cells are extremely diverse. We have many different types of cells in our bodies, ranging from our heart cells all the way to our brain cells, skin cells, and these different types of cells, they have different structures which are going to influence the different functions. So all cells, or nearly all cells, are microscopic, so we require a microscope to see the cells. Two types of microscopes are the light microscope and the electron microscope. The light microscope is going to be using light, whereas the electron microscope uses electrons. The light microscope is going to have a lower resolution and magnification than the electron microscopes, but the advantage is the ability to see the dynamic movement of uh, intracellular structures within the cell. So we see right here the different micrographs, pictures of uh, specimens under the microscope, and we see different scientists using the electron microscope in the top picture, and also a scientist using the compound light microscope in the bottom left. And here we see a chart of a range of smallest to largest from the left to the right. And also this chart shows you kind of, or this range shows you which organisms or molecules require which microscope. And as you can see further to the right, we have the larger organisms that can be seen with the unaided eye. So cells are microscopic, so we need to use a microscope. And also, small cells have a advantage. When we have several small cells versus just one big cell, we can maximize our surface potential. And this is an adaptation that's useful for example, the maximum absorption of the small intestine. So here we see kind of a representation of that. If we had one 4 centimeter cube, one big cube versus 60 for 1 centimeter cubes, the surface area of the 64 1 centimeter cubes would be much greater than just the single 1 4 centimeter cube, even though their volumes are equal to each other. Okay, so next we're going to discuss the plasma membrane. So the plasma membrane is also called the cell membrane, and the plasma membrane is made up of phospholipids. And there are going to be two phospholipid layers, so we call this a phospholipid bilayer. The plasma membrane, so therefore, right, we have like three different names for the cell membrane, which is the cell membrane itself, the plasma membrane, or we can refer to as the phospholipid bilayer. So each phospholipid molecule has a polar head and nonpolar tails. Another word for polar heads is going to be hydrophilic heads, and for the nonpolar tails, they're going to be hydrophobic. So here we see a representation of the cell membrane. So we can see on the top left right here, one phospholipid molecule. So one phospholipid is going to be composed of our polar head and two nonpolar fatty acid tails. So if you look at the cell membrane right here, the polar heads are going to be facing the fluid on the outside of the cell. And the polar heads in the other layer of the phospholipids is going to be facing the fluid in the inside of the cell. So that is why the polar heads in red are going to be called polar. because they're also going to be hydrophilic, water-loving, versus the hydrophobic or nonpolar tails that are going to be facing away from the water, and they're kind of forming this core right here, the core of the cell membrane. And lastly, you notice these proteins that are embedded in the plasma membrane, which are going to have several different functions. They can act as transport proteins to transport materials across the cell membrane, or they can be enzymatic proteins. They can be used for cell communication, and etc. So let's take a look at the different classifications of proteins that can be embedded in the cell membrane. So we have channel proteins, which are going to be important for transferring or transporting certain molecules across the cell membrane. So the channels are like tunnels for specific molecules. An example of a channel protein would be aquaporin, which is going to transport water across the cell membrane. Okay, so again, like I mentioned earlier, transport proteins are involved in the transport or passage of molecules through the membrane. And sometimes this is going to require energy, and sometimes it doesn't require energy. And if you look at this transport protein, it doesn't look like a channel. So in this case, this is called a carrier protein, carrier transport protein. So we can have different types of transport proteins. Next, we have cell recognition proteins. So this enables our body to distinguish between our own cells and cells of other organisms. So this is important because we don't want our cells to attack our own body cells. We don't want our immune system to attack itself, which would be an autoimmune response. Receptor proteins are important for receiving certain signal molecules from other cells or other parts of the body. And this is important because it has to do with cellular communication, and also it's going to produce a certain intracellular or inside the cell response that's important for the development of the cell and also other aspects of the cell. Enzymatic proteins are important for metabolic reactions. For example, in the mitochondrial membrane, we have an enzymatic protein called ATP synthase, which is important for producing ATP, which is the energy currency of the cell. Junction proteins are going to form the junctions between the cells. This can be important for adhesion, right? So sticking one cell to another cell, holding them fast together, and also communication between cells. The cell theory states that all organisms or all living things are composed of cells. And also another point from the cell theory is that all cells come from pre-existing cells. So cells cannot just appear spontaneously out of nowhere. They all originate from a cell that already existed. All cells have a plasma membrane, a cytoplasm, and also genetic material. So we have two classifications of cells that all organisms are going to be under, either or. So we have prokaryotic cells and eukaryotic cells. So prokaryotic, that word right here, prokaryotic means without nucleus, and eukaryotic means true nucleus. So the eukaryotic cells do have a nucleus. We'll take a look at the prokaryotic cell first. So prokaryotic cells are going to include organisms from the domain bacteria and archaea. Prokaryotic cells are smaller than eukaryotic cells, and bacteria, being prokaryotic cells, are well known because some of them cause disease. Some bacteria are used to manufacture chemicals, foods, drugs, and so on. And some bacteria are also good for you. There's some good bacteria, such as probiotics, and also there's bacteria in our gut or our digestive system that are beneficial to our body's function. So let's take a look at bacterial structure. So generally, the bacteria has a cytoplasm that's surrounded by a cell membrane, and also the bacteria has a cell wall just outside of the cell membrane. And sometimes bacteria have a capsule, which is a protective layer. So the bacteria have a cell membrane, and that's the same as eukaryotes. So that's important to remember. The cell wall of the bacteria is going to help maintain the shape of the bacteria. And a major difference in the shape of the DNA for bacteria is that bacteria have circular DNA, unlike the linear DNA of eukaryotic cells. Bacteria also have ribosomes because they need to make proteins. And bacteria also have appendages that are going to help them either attach to surfaces or just to travel. So it helps with motility and also DNA transfer. So the flagella is used for propulsion or motility. The fimbriae are used by the bacteria to attach to surfaces, and the conjugation pili is used for the bacteria to transfer their DNA to other bacteria. So this is what a bacterial cell looks like. And as you can see, we have the different appendages. We have the fimbriae, the little hair-like structures coming out of the bacteria, and also the flagella. That's going to help with the bacterial motility. Next, we'll take a look at the eukaryotic cell. So the eukaryotic cell is going to include protists, fungi, plants, and animals. So if you are not classified as a bacteria or an archaea, then you are going to be a eukaryotic cell. Eukaryotic cells are much larger than prokaryotic cells, and eukaryotic cells have these structures called organelles. So organelles are structures inside of the cell that are surrounded by a membrane. Another way to say that is that organelles are membrane-bound structures. There are 4 categories of organelles. We have the nucleus and ribosomes, the organelles of the endomembrane system, the organelles that are energy related, and the organelles that have something to do with the cytoskeleton. So here's a look at the structure of a typical animal cell, which is a eukaryotic cell. And if we go back and compare this to the structure of the prokaryotic cell, you can immediately recognize that the eukaryotic cell is much more complex. And that is because it has several more organelles, a diverse amount of organelles. So here's the structure of a plant cell. So this is going to be an actual micrograph or a picture under a microscope. And the plant cell is also a eukaryotic cell. So plant cells are going to have a chloroplast. That's going to be different from the animal cells because the chloroplast is where photosynthesis occurs. So here we see a kind of diagram of the plant cell. And you can notice the difference in the shape immediately. The animal cell is more roundish, round shaped, whereas the plant cell is more boxy or rectangularly shaped or square shaped. And you also notice the huge central vacuole here. That's a lot bigger than the animal cell vacuole, which stores water and other cellular material. So we'll move on to the organelles. So let's talk about our different groups of organelles. So we have the nucleus and the ribosomes. To avoid any confusion, I wouldn't have grouped them together like this. So I just want to let you know that ribosomes are not organelles. Ribosomes are not surrounded by a membrane. They're not membrane bound, so ribosomes are not organelles. However, the nucleus is a organelle. So what is the function or the job of the nucleus? The nucleus is the storage place for DNA and also RNA after we go through transcription. The nucleus contains the chromatin, which is the DNA wrapped around these proteins called histones. And we need chromatin because we need to fit the long strands of DNA into the tiny nucleus. So we need to wrap the DNA around the histone proteins in order to fit the DNA in the nucleus in the form of chromatin. DNA is organized into genes, and genes are going to be basically just sections of the DNA that's going to code for or express, or in this case specify, a certain polypeptide or a protein. And so the information from the DNA ultimately is sent to the ribosome in the form of mRNA, messenger RNA. And the ribosomes are going to read the message from the mRNA to create the amino acid sequence or the polypeptide. The nucleolus is also located inside of the nucleus, and this is the region where ribosomal RNA or rRNA is made. The nucleus is surrounded by a nuclear envelope, which is a double membrane, and hence this is why the nucleus is considered an organelle. It's bound by a membrane. The nucleus also has these tiny holes inside of them called nuclear pores, which allow certain materials to go in and out of the nucleus. So here's a look at the structure of the nucleus. So we see here the nucleus is surrounded by the nuclear envelope. And when we look inside of the nucleus, we see the nucleolus right here, the smaller ball or circle. Next, we'll discuss ribosomes. So ribosomes are not organelles. But what is the job of the ribosomes? The ribosome's job is to manufacture proteins. Another word for that is to synthesize proteins or protein synthesis. So this happens in the cytoplasm of the cell, or it can happen near the rough endoplasmic reticulum, which has the ribosomes attached to it. So ribosomes are going to be found in both prokaryotic and eukaryotic cells. And this makes sense because ribosomes are the protein factory of the cell. And both prokaryotes and eukaryotes need proteins in order to build essential parts of itself or its own structure and architecture. The ribosome is composed of two parts or two subunits, and the ribosome is a mix of proteins and ribosomal RNA called rRNA. So how do the ribosomes know what kind of protein to manufacture or synthesize? The ribosomes need to first receive an instruction manual called mRNA, messenger RNA. Once it does, the ribosome knows what kind of sequence of amino acids to build in order to build our polypeptide. And remember, students, amino acids are the building blocks of polypeptides. And polypeptides can also be just called proteins. In eukaryotes, the ribosomes are located either in the cytoplasm or they're attached to the endoplasmic reticulum. More specifically, the rough endoplasmic reticulum. The ribosomes in the cytoplasm are called free ribosomes, and the ribosomes in the endoplasmic reticulum are attached ribosomes. So here we see the nucleus, and also we see the rough endoplasmic reticulum right here, along with the ribosomes in red. If you follow the sequence from 1 to 4, we can see how mRNA is first transcribed from DNA, and the mRNA next has to leave the nucleus through the nuclear pore. And now that the mRNA is in the cytoplasm, the mRNA has to go to a ribosome. And the ribosome is going to read the mRNA in order to produce a polypeptide, which is going to be the, I believe, the blue strand right here. Next, we're going to talk about the endomembrane system. So the endomembrane system is going to be made up of the nuclear envelope of the nucleus, the membranes of the endoplasmic reticulum. So we have the rough endoplasmic reticulum and also the smooth endoplasmic reticulum, the Golgi apparatus, also called the Golgi body, and numerous vesicles. And vesicles or transport vesicles are going to be these sacs of membranes that are going to store and package certain cellular material or molecules from one area of the cell to another area in the endomembrane system. So the endomembrane system is going to help compartmentalize the cell, help to organize the cell into different compartments. So this is going to allow the restriction of certain reactions, chemical reactions, to occur in specific regions. And these chemical reactions might be bad for the cell. The first component of the endomembrane system is the endoplasmic reticulum. The endoplasmic reticulum is a system of membranous channels and saccules, and they are physically continuous with the outer membrane of the nuclear envelope, which is part of the nucleus. So you're going to see the endoplasmic reticulum right next to the nucleus. The rough endoplasmic reticulum, also called the rough ER, is going to be rough because it has ribosomes attached to it. Also, the rough ER is going to modify proteins in the empty space of the rough ER, which is called the lumen. So the lumen is the hollow space within the rough ER. And modify proteins can also mean it helps to fold the proteins. And that's important because when the proteins are first made, the proteins are not finished. We need to modify them and further fold them in order to produce a final protein product. The rough ER also forms the transport vesicles, which are important for transporting certain cellular materials or molecules. And these transport vesicles are going to send the proteins to the Golgi apparatus. The smooth ER is also continuous with the rough ER. However, the smooth ER is smooth because it does not have ribosomes. The smooth ER is going to be involved in manufacturing or synthesizing lipids such as phospholipids and steroids. So the function of the smooth ER is going to depend on the cell. So in some cells, it can produce testosterone, and in other cells, such as liver cells, it can detoxify drugs. So here's a picture of the rough endoplasmic reticulum that's studded with ribosomes versus the smooth endoplasmic reticulum that does not have ribosomes. Both are continuous with one another. So again, we see that picture here, the rough endoplasmic reticulum and the smooth, so the rough endoplasmic reticulum and the smooth endoplasmic reticulum here. And right below it, we see the Golgi apparatus. So the rough endoplasmic reticulum is going to send its proteins in these sacs called transport vesicles, which are going to be transported to the Golgi apparatus. And you can kind of see how the transport vesicle is going to be fusing out of the rough ER, and it fuses into the Golgi apparatus. And this makes sense because the transport vesicles are made of membrane. So what does the Golgi apparatus do? What does the Golgi apparatus do? It further modifies the protein or lipid, and it packages them into vesicles once again. And these secretory vesicles from the Golgi apparatus is going to be sent to wherever the cell needs those proteins. Or it can be sent outside of the cell through exocytosis. So what is the job of the Golgi apparatus? As we saw earlier, the job of the Golgi apparatus is to further modify cellular molecules or materials and package them and send them out to whichever part of the cell needs those proteins or those molecules. So the Golgi apparatus is composed of a stack of flattened saccules, just flattened cell membranes or membranes. And this is what's known as the transfer station. It's a kind of like the USPS, like the mailing building. It receives the vesicles from the rough endoplasmic reticulum or the smooth endoplasmic reticulum. It further modifies them, and it sorts them and packages them or repackages them for a new destination in the cell. So that new destination can actually go to the lysosomes. And the lysosomes are vesicles that digest bigger molecules and breaks it down into smaller pieces. So the lysosome is kind of like the recycle truck of the cell. It digests wastes, and it also digests big pieces of food molecules. So it does this because the lysosomes contain a digestive enzyme called catalase, which breaks down the wastes and the food molecules into smaller pieces. Next, we have vacuoles. Vacuoles are going to be important for storage within the cell, and also they can get rid of cellular water or excess water in the cell. Vacuoles can be important for storing water and other important nutrients of the cell. And particularly for the plant cells, they have a very large central vacuole that's important for the storage of water. So here we see vacuoles doing different things or having different functions in different types of cells. So on the left, we see a paramecium, a single-celled organism that has contractile vacuoles, which help the paramecium to get rid of excess water inside of the cell. On the right, we have a plant cell with a huge central vacuole here that's going to store water and other important cellular material. Next, we're going to move on to the group of organelles related to energy. So the mighty mitochondria is going to be the powerhouse of the cell, and it's also a organelle because it is going to be surrounded by a membrane. And actually, it's going to be a double-membraned organelle. Mitochondria are going to be found in both plants and animals because plants and animals both require the energy called ATP that the cells need in order to function. Usually mitochondria are only visible under an electron microscope. And like I said earlier, they have a double membrane. So what do the mitochondria do? They break down carbohydrates, so glucose in particular, and through that process, they produce energy in the form of ATP. And that stands for adenosine triphosphate. So this whole process of breaking down carbohydrates in order to basically get ATP is going to be part of a process called cellular respiration. And in cellular respiration, the cell is going to need oxygen, and a byproduct of cellular respiration is carbon dioxide. And another way to look at cellular respiration is that it's the opposite of photosynthesis. Photosynthesis is going to use sunlight and CO2 and water to produce glucose. Whereas in cellular respiration, we use glucose and oxygen in order to break down the glucose into other constituent molecules in order to get ATP. The inner membrane of the mitochondria, it folds into what we call cristae, and this increases the surface area of the mitochondria. The core of the mitochondria has this fluid called the matrix, and this is a mixture of enzymes assisting in the breakdown of our carbohydrate, which is glucose. And this is eventually breaking down that carbohydrate or the sugar is going to result in ATP synthesis or the production of ATP. The matrix of the mitochondria is also going to contain its own mitochondrial DNA and ribosomes, which is very interesting how an organelle can have its own DNA. Chloroplasts are going to be only found in plant cells and other photosynthetic organisms, which can include protists, and algae are protists. So chloroplasts uses the solar energy from the sunlight to synthesize or create carbohydrates, glucose, through the process of photosynthesis. So for chloroplast, they're going to have three membranes. So we have a double membrane enclosing the fluid of the chloroplast called the stroma. And the thylakoids are going to be the innermost membrane, or the third membrane, and they're going to be arranged in a sort of like a disc. And it's the thylakoid membranes that actually contain the pigment called chlorophyll that captures the sunlight energy. Chloroplasts also have their own chloroplast DNA and ribosomes as well. So you can see here we zoomed in on a chloroplast, and it has a double membrane. On the outside it's clear, which is enclosing the thylakoids, which are the green stacks of membranes. They kind of look like pancakes. So they're going to be stacks of membranes, and they're going to be inside the stroma. So the stroma is just the space that's filling the remainder of the chloroplast on the inside. So here's a micrograph of the chloroplast. And here is what the mitochondria looks like. Mitochondrion. So we see a outer layer and an inner layer, and we can see the inner layer folds, and these folds are called cristae, and the inside, or the core of the mitochondria, is going to be the matrix. Next, we will discuss the cytoskeleton and how they work with the motor proteins in the cell. The cytoskeleton, so cyto, if we look at the word, it means cell. So cytoskeleton just means cell skeleton. And it's a network of interconnected protein filaments and tubules. So the cytoskeleton is made up of proteins, and they can be filaments or tubules. They extend from the nucleus to the cell membrane, and the cytoskeleton is only in the eukaryotic cells. So just like our skeletons, they help to maintain the shape of the cell, and the motor proteins work with the cytoskeleton to allow cell movement and movement of the organelles. So examples of motor proteins include myosin, kinesin, and dynein. So motor proteins. Motor proteins work with the cytoskeleton to allow cellular movement. So myosin is an important motor protein that is found in muscular cells. They interact with the cytoskeleton called actin, which is also called a microfilament, in order to produce muscle contraction in humans and other animals. And also myosin is found in certain cells to allow them to move. We have the motor proteins kinesin and dynein, which move along the microtubules, which is part of the cytoskeleton, and this allows for transport vesicles from the Golgi apparatus to travel to its final destination. So here's an example of myosin interacting with the actin filament, and the actin filament is part of the cytoskeleton, while the myosin is a motor protein. And you can see how both of them are going to interact together in order to produce muscle contraction. So when the head of the myosin filament pulls on the actin filament, this is actually what happens during muscle contraction, which is the shortening of the muscle. And here we have the motor proteins kinesin, and the kinesin is going to travel along the microtubule, and the microtubule is the cytoskeleton in this case. And you can see how the microtubule is kind of like a road that the kinesin motor proteins are going to travel on. And notice what the kinesin motor proteins are carrying with them at the top. So those red circles here are going to be the vesicles. the transport vesicles which contain the cellular material that must be transported to different parts of the cell. Continuing with the cytoskeleton is made up of 3 different types of proteins. So we have the microtubules, the intermediate filaments, and the microfilaments. The microtubules are small hollow cylinders, so they look like tubes, and their assembly is controlled by centrosomes, which are located in the cell. They help maintain the shape of the cell and act as a kind of track or road for the organelles and other vesicles to move on. As we saw earlier with kinesin, intermediate filaments are intermediate in size. So microtubules are the biggest of the bunch. Intermediate filaments are in the middle, and microfilaments are the smallest proteins of the cytoskeleton. Intermediate filaments are going to be in a rope-like assembly, and they run from the nuclear envelope of the nucleus to the cell membrane. So here is a picture of microtubules. And you can kind of see how they're coming from the centrosome right there. And so this is what I mentioned earlier. So this is our 3rd type of cytoskeleton, the actin filaments. So the actin filaments are also called the microfilaments. So actin filaments are also called microfilaments. So these are going to be the smallest proteins of the cytoskeleton. They are 2 chains of monomers twisted into a helix, and they form a dense web to support the cell. Okay, so we move on to what centrioles are. So centrioles are going to be located inside of centrosomes, and the centrioles are made up of 9 sets of microtubules. Microtubule triplets. So centrioles are made up of microtubules. So 2 centrioles lie at right angles, and centrioles are only located in animal cells and not in plant cells. And this has to do with the differences in cell division. So the centrioles right here are important in cell division. They are made up of microtubules and triplets of microtubules arranged in this specific pattern. And now we will move on to cilia and flagella. So we've so far talked about the endomembrane system, which was composed of several different organelles working together to transport cellular material. And also we talked about the cytoskeleton, which was important for maintaining the shape of the cell, also for transporting organelles in different vesicles, and also cell division. And so the cilia and flagella are important for movement, and they're also important for other functions. So in eukaryotes, the flagella is helpful for cell motility, so the movement of the cell. So there's similar construction in both the cilia and flagella. So they are going to be made up of microtubules, which is going to be the cytoskeleton, one of the proteins of the cytoskeleton. The cilia is shorter and more numerous than the flagella. So the cilia and flagella are also important for the movement of the cell or fluids past the cell in eukaryotic organisms. So here are some examples of both cilia and flagella. So we can see flagella in the sperm cells of male animals and humans, which help with the movement or the motility of the sperm cell. And we can see the cilia in the bronchial wall of lungs. We can see cilia in the those cells in the bronchial walls of the lungs. And the job of the cilia is to trap any dust or any other particles that might get into the lungs. And you can see on the right here, we have the arrangement of the flagella right here, or flagellum, which is made out of microtubules, and they're going to be arranged in a certain pattern right here. Next, we are going to discuss the outside of the eukaryotic cell. And when we take a look at plant cells, they have a cell wall. So plant cells have a primary cell wall, which is going to be made up of cellulose and also non-cellulose substances. And the cell wall actually can stretch when the cell is growing. And in some plants, and not all plants have this, but in some plant cells, they have a secondary cell wall, which forms inside the primary cell wall. And so this can be found in woody plants. And this, in the secondary cell wall, we can see a protein called lignin, which adds strength. The plasmodesmata is a cell junction, and it's a channel that's going to connect two plant cells to each other so that they can exchange water and small solutes. And so if we look right outside the eukaryotic cell, whether it be an animal cell in this case or a plant cell, so in this animal cell, right outside the cell, we have something called an extracellular matrix. So this extracellular matrix is actually going to determine the quality of the tissue of our of a certain part of the body. And the extracellular matrix is going to be made up of water and other proteins such as elastic fiber proteins, elastin, collagen proteins. Also, we have polysaccharides, these sugars. And depending on the composition of the extracellular matrix, this is going to affect the quality of the tissue. So for example, if you look at bone tissue versus blood tissue has a lot more water in it than bone tissue, which has a lot more, let's say, calcium deposits and also collagen proteins in it. So if you look at animal cells versus plant cells, animal cells do not have a cell wall. Their extracellular matrix is made up of fibrous proteins and polysaccharides. Collagen and elastin proteins are well-known proteins in the extracellular matrix of animal cells. And the matrix, like I said earlier, varies. So in cartilage tissue, it's going to be, the matrix is more flexible, and in bone tissue, the matrix is going to be a lot harder. The cell wall is not only found in plant cells, but also in fungi cells and also protist cells. So the extracellular matrix, again, found in animal cells, is a meshwork or a network of fibrous proteins and polypeptides in close association with the cell that produce them. So the extracellular matrix can have different ratios of collagen or elastin. Collagen is a protein that helps to resist stretching and giving the tissue its tensile strength. And elastin is a protein in the extracellular matrix that provides resilience to the tissue, and it allows the tissue to stretch. Lastly, we will discuss junctions between cells. So a junction is just going to be the area between two cells. So right here, this would be the junction, and we have the two cells right here that are interacting with one another. So a cell junction is going to have proteins in the junction that's going to influence the way that the two cells interact with one another. So a there are three different types of junctions: adhesion junction, tight junction, and gap junctions. The adhesion junction is going to help one cell and another cell to adhere to each other, to hold fast to each other through these intercellular filament proteins. When we look at the tight junction, this is going to, the tight junction is going to include these other proteins called occludin proteins that are going to seal the two cells shut. They're going to hold the two cells fast to each other to prevent leakage in the tissue. So an example of tight junctions is in your urinary bladder cells. So that prevents any urine from leaking out of your bladder. And then we have the gap junctions. So the gap junctions are going to have protein channels going across the junction from one cell to the other, which allows the transport of materials between the two cells. So again, taking a closer look right here, the adhesion junction is going to contain these intercellular filaments that's going to join the two cells together tightly. And next we have the tight junctions, which is going to include proteins that are going to create an impermeable barrier and prevent any leakage in the tissue. And then we have the gap junctions, which look more straightforward. We have protein channels in the junctions that allows the transport of materials between the two cells.